Welcome everyone, Mike Fave here. Today I'm going to go through what exactly is the Randle cycle. So on the most recent Bioenergetic Healthline number 8, Jay, Danny, and I were discussing some of the issues with Bart K's description of the Randle cycle. And we didn't really dive into the specific mechanisms of the Randle cycle. We kind of glossed over and discussed them, or more specifically discussed the parts where Bart was at least incorrect from our perspective on the Randle cycle. So I wanted to take you guys through today the actual mechanisms of the Randle cycle, go through some graphics, show you how carb oxidation blocks fat oxidation. I'm going to start out by using a research article here, The Randle Cycle Revisited, A New Head for an Old Hat. That's the title of the article. And I'm going to show you some graphics just so we can break down and you can actually see what is going on, at least theoretically, in the mitochondria with carb oxidation being blocked by fatty acid oxidation. So we're going to start off here. I'm not going to read every single element of the article. I'm going to pull out the relevant points. And what we're going to initially discuss is what exactly is the Randle cycle. And there's a good sentence here. And basically, the Randle cycle can be described by inhibition of glucose utilization by fatty acids. It is a form of glucose intolerance that resembles or may lead to insulin resistance, i.e. the impaired capacity of insulin to increase glucose uptake by muscle and adipose tissue, accompanied by increased lipolysis and increased hepatic glucose production. So, quick translation here. Fatty acid oxidation at the cellular level blocks glucose utilization by those same cells that are burning fatty acids, burning in quotations. This leads to a form of glucose intolerance that could be a feature of insulin resistance in pathologic situations. And essentially, you also will see increased lipolysis or releasing of fatty acids into the bloodstream and increased production of liver glucose, or the production of glucose by the liver in this state. So there's kind of a lot going on here, but the key point is that fatty acid oxidation blocks glucose utilization or glucose oxidation at the cell. That's essentially the Randle cycle. There is the opposite effect where glucose oxidation can block fatty acid oxidation to some extent, but the most major piece here to discuss is the inhibition of glucose utilization by fatty acid oxidation. That's the main point that I'm going to talk about here today, because I think it is one of the most important ones in this whole carb versus fat debate that we're seeing with the keto, carnivore, paleo, bioenergetic spheres, etc. So let's actually dive into the mechanisms. So basically they continue here, and what they're talking about in this section is that glucose is spared and rerouted during fatty acid oxidation. And this is actually a survival mechanism when you don't have adequate carbohydrate, glucose, other forms of carbohydrates such as fructose coming in from the diet or fatty acids. And what winds up happening is that this process basically inhibits glucose oxidation and spares that glucose for the central nervous system and other tissues that directly rely on carbohydrate. That could be the gonads, adrenal glands, red blood cells, but specifically the central nervous system. That's the main one to discuss, and that it requires a decent amount of glucose on a regular basis. So it is absolutely essential that the body has glucose for the central nervous system. So the rest of the body will essentially run on fatty acids to allow the central nervous system to oxidize that glucose. So what they say here is, by inhibiting glucose oxidation, fatty acids and ketone bodies so contribute to a glucose sparing effect, an essential survival mechanism for the brain during starvation. So this is essentially what I just described. When you don't have enough glucose on board, fatty acid oxidation takes precedent in the vast majority of tissues, sparing that glucose for the central nervous system, which absolutely requires glucose to function appropriately. Even with ketone production, which the central nervous system can use ketones, there's still a large percentage of glucose oxidation that will occur even with ketones present. So the ketones are a survival mechanism as well, as is the rest of the body using fatty acids and sparing that glucose for the tissues that need it. So they continue further here. They say, the glucose fatty acid cycle is not restricted to the fasting state and is readily observed in the fed state after a high-fat meal or during exercise when plasma concentrations of fatty acids or ketone bodies increase. Under these conditions, Part of the glucose that is not oxidized is rerouted to glycogen, which may explain the rapid resynthesis of muscle glycogen after exercise. Rerouting of glucose towards glycogen also explains the increased glycogen content in muscles found in starvation or diabetes. So something important that's kind of implied here is that during fasting, when you have a lot of lipolysis or release of free fatty acids because you don't have adequate glucose coming in exogenously, the body will essentially upregulate fatty acid oxidation, and this will spare some glucose as well. So it's not just eating a high-fat meal that can lead to increased fatty acid oxidation. States such as fasting, where you have to rely on your body stores of nutrient or substrate, fats and carbohydrates, states where you've used a large portion of your, or you've used a decent portion of your carbohydrate readily available and have started to undergo lipolysis, like exercise. Another state would be stress, would increase all of these states would increase plasma fatty acids, and those plasma fatty acids could impair glucose oxidation or essentially spare glucose oxidation. Something that they talk about here that's a little bit tangential to the overall picture that we're talking about is that in states where there's heavy amount of lipolysis and fatty acid oxidation, that glucose won't necessarily be oxidized. So it actually, again, it gets spared and it can be rerouted towards glycogen synthesis since it's not being used. And so you can find this situation in starvation or in diabetes. Both states are characterized by high amounts of fatty acid oxidation with impaired, surprisingly in starvation, with impaired insulin sensitivity to some extent because the body is releasing so much fatty acids and oxidizing so much fatty acids that, as we discussed above up here, that will inhibit glucose utilization and can lead to insulin resistance. Now, insulin resistance, you can break it down. It's not a perfect, it gets a little sticky, but you can break it down in terms of physiologic insulin resistance and pathophysiologic insulin resistance. So pathophysiologic insulin resistance is what you see in diabetes. It's what you see in states where you basically can eat carbohydrate, but you can't effectively oxidize it, versus a non-pathologic or a non-pathogenic insulin resistance. What's the word I'm looking for here? A physiologic insulin resistance. In a physiologic insulin resistance, this occurs for example, in like a high-fat diet where you are indeed insulin resistant, but it's a function of not having adequate carbohydrate and having a large amount of fatty acids flooding the system postprandially or after you're eating. And this essentially leads to excess fatty acid oxidation that blocks glucose oxidation. Now, in people who are doing high-fat diets or keto diets or very low carbohydrate diets, when they add carbohydrate back in, they can then again become insulin resistant. It just takes a period of time. However, with diabetics or people with metabolic syndrome, what you wind up seeing is even if they have carbohydrate present, they still have insulin resistance. And there's a couple different things going on in these different states. There is some congruency between them. So there is some elements that are similar or some metabolic functions that are similar in both states, but they're not exactly the same. There is a difference. Now, that doesn't mean that over time, an excess of fatty acid oxidation could lead to issues with glucose oxidation in the long run. That's definitely possible. But in most situations that I think we'd be talking about with somebody who's on keto or carnivore, if they switch to a higher carb diet appropriately, again, the key word here is appropriately, not I found the bioenergetic way of eating and I slammed a whole bunch of ice cream and orange juice and sugared milk and I gained a bunch of weight. I think there's a better way to introduce those things, but when they do things appropriately, you're able to improve or recover that insulin sensitivity by minimizing an excess of fatty acid oxidation. Also, again, I just want to point this out specifically. Stress states, being under large amounts of stress, triggers lipolysis and gluconeogenesis and fatty acid oxidation through cortisol, adrenaline, glucagon, growth hormone. And in these situations, you have insulin resistance triggered by those stress hormones. You see the same thing in diabetes, and these same hormones are also strongly upregulated in keto and carnivore and other very low carb situations, and even in fasting situations. Even intermittent fasting, they're showing decreases in thyroid hormone elevations in glucocorticoids chronically or over with chronic fasting, especially depending on how long those fasts are. Upregulations of the catecholamines, upregulations of growth hormone. These are actually touted as benefits in the fasting communities, and then decreases in androgens and other sex steroids. So those hormones are elevating not because for example with the growth hormone in those situations is elevating not to like grow your tissues or help you lose weight. It's increasing during fasting situations to help you oxidize the fatty acids better that you're liberating from your tissues. So it's not necessarily a good thing. You have to understand the context of why these hormones are elevating. Anyway, I know I got off on a tangent. Let's continue. Here, this is where we get specifically to the carbohydrate or fatty acid oxidation blocking carbohydrate oxidation and what exactly the mechanisms are. So I'm going to read through this section, this little pink area I'll read after, and then I'm going to go through the graphs. So I'll read through this, go through the graphs of things or the diagrams, and then I'll get to this section after to kind of cement the point home, and that'll wrap up the video for us today. So what they start here is they say, Randle, so this is Sir Philip Randle, demonstrated that impairment of glucose metabolism by fatty acid or ketone body oxidation was mediated by a short-term inhibition of several glycolytic steps, namely glucose transport and phosphorylation, 6-phosphofructose-1 kinase, so PFK1, and pyruvate dehydrogenase, so PDH. So the first one that fatty acid oxidation inhibits, well, it's not the first one, but the first one mentioned here is glucose transport and phosphorylation into the cell. So when glucose gets to the cell, it gets converted to glucose 6-phosphate. This step gets impaired by fatty acid oxidation, but it's not the major area that gets impaired. The next one is 6-phosphofructose-1 kinase. So this is an enzyme in the glycolytic pathway, an enzyme in the process of glycolysis. So this gets blocked to some extent, but again, this isn't the major enzyme that gets blocked by fatty acid oxidation. Now, pyruvate dehydrogenase is the major enzyme that gets blocked by fatty acid oxidation. And this enzyme is extremely important because this is the enzyme that links glycolysis. So that's the process of taking glucose and creating 2-pyruvate to the Krebs cycle, where that pyruvate is converted to acetyl-CoA and eventually a citrate. And then from there, it runs through the Krebs cycle. You build up NADH and FADH2, which are electron carriers. They're the reduced forms of NAD plus and FAD, respectively. So the reduced meaning 2 electrons have been added, or not 2, but electrons have been added to them. Those electrons have a negative charge, which is why they're called reduced. And then essentially those get moved to the electron transport chain. And when they're moved to the electron transport chain, they move through the electron transport chain enzymes. The enzymes pull the electrons off those carriers, and in doing so, pump protons or H plus ions into the inner mitochondrial space, which creates a gradient. And then that gradient of protons runs through an enzyme called ATPase, and it's kind of like a hydroelectric dam, and then it produces ATP. And I'm going to go over all of this in just a second. So if everything I just, with a graphic, so if everything I just said sounded like a whole bunch of non-English or just gibberish to anybody, then don't worry, I will show you exactly what I mean. But the main thing to keep in mind here is that PDH, pyruvate dehydrogenase, is the main enzyme that gets blocked by fatty acid oxidation. Phosphofructose kinase and the glucose transport and phosphorylation, both of those are secondary to PDH. PDH is really the main one. So what they say is the extent of inhibition is graded and increases along the glycolytic pathway, being most severe at the level of PDH and less severe at the level of glucose uptake and phosphofructose kinase. So this is exactly what you're saying. Pyruvate dehydrogenase is the main enzyme that gets blocked. This sequence occurs because the initial event triggered by fatty acid oxidation is an increase in the mitochondrial ratios of acetyl-CoA to CoA and NADH to NAD+. So you get a buildup of these reduced carriers. This is what Dr. Pete is talking about when he talks about reductive stress. So you're seeing a buildup of this NADH in ratio to NAD. And we'll see why that's important in just a second. Excuse me. So, and again, both of the excess acetyl-CoA in ratio to CoA and an excess of NADH in ratio to NAD+, both inhibit pyruvate dehydrogenase activity. So these are some of the main mechanisms. There's other that we'll talk about here. Then they say it has been proposed that these changes lead to an accumulation of cytosolic citrate, which in turn inhibits phosphofructose kinase, followed by an increase in glucose 6-phosphate, which eventually inhibits hexokinase. So hexokinase is that enzyme that takes the glucose, helps to create glucose 6-phosphate and bring that glucose into the cell. However, a buildup of glucose 6-phosphate, basically a buildup of the product of this reaction, inhibits the reaction to some extent. So, and again, they talk a little bit about this in more detail, but this is not as important as pyruvate dehydrogenase. And then citrate, which is a mediator, or it's a mediator, a metabolic intermediate in the Krebs cycle, inhibits phosphofructose kinase 1. And so the inhibition, so the citrate builds up because of the poor activity of PDH in the Krebs cycle, and then that buildup of, and again the poor activity of PDH in the Krebs cycle happens because of some issues with the buildup of NADH and acetyl-CoA. And then essentially what you wind up see happening is the citrate basically goes onto the phosphofructose kinase 1 enzyme and impairs its function. So this is a specific mechanism where the buildup of citrate is impairing this particular enzyme here. And again, phosphofructose kinase 1 and glucose and a hexokinase or a buildup of glucose 6-phosphate aren't the main problems with fatty acid oxidation blocking carb oxidation. It's really that blockage at pyruvate dehydrogenase. So, and again, I'm going to show you guys this whole pathway in just a second. There's more blocks on the pyruvate dehydrogenase enzyme, and we're going to get into that right now. So what they say is the products of PDH exert a feedback inhibition on its activity. So the product of PDH is acetyl-CoA, and essentially what happens is when that builds up, it inhibits PDH's ability to produce more acetyl-CoA. So it has an inhibitory effect on PDH. And this is, in chemistry, this is kind of known, I think it's Le Chatelier's principle, where a buildup of the product of a metabolic reaction can inhibit the reaction to some extent or push the reaction in the other direction if it is reversible. And there's a couple reasons for why, but essentially the buildup of the products of pyruvate dehydrogenase block its activity. So if you build up of that, the enzyme stops functioning as well in conjunction with a buildup of the acetyl-CoA NADH, which is actually part of this mechanism anyway. What they also talk about here is dedicated mitochondrial kinases, pyruvate dehydrogenase kinase, phosphorylate and inactivate pyruvate dehydrogenase, whereas pyruvate dehydrogenase phosphatases have the opposite effect. Control of pyruvate dehydrogenase activity depends on the relative activities of PDK and PDP and on the extent of phosphorylation of the E1 subunit sites. So PDK is the pyruvate dehydrogenase kinase, and PDP is the pyruvate dehydrogenase phosphatase. Now, phosphatases increase pyruvate dehydrogenase activity, whereas kinases decrease pyruvate dehydrogenase activity. So if you have an upregulation of pyruvate dehydrogenase kinases, then pyruvate dehydrogenase gets shut down essentially, and that impairs the flow of carbohydrate from the glycolysis into the Krebs cycle. And then if you have increased activity of pyruvate dehydrogenase phosphatase, then you will increase pyruvate dehydrogenase activity. Also, if pyruvate dehydrogenase kinase activity gets downregulated, then pyruvate dehydrogenase activity can increase. So though all of those mechanisms are at play, and that'll become important as we talk here. So what they say is pyruvate dehydrogenase substrates and products also control pyruvate dehydrogenase kinase activity. Pyruvate, or its analog dichloroacetate, inhibits, whereas acetyl-CoA and NADH stimulate pyruvate dehydrogenase kinase with isoform-specific differences in sensitivity. So depending on the type of pyruvate dehydrogenase, the isoform, the different type of enzyme, some are more sensitive than others to these products, acetyl-CoA or NADH or pyruvate or a similar compound to pyruvate dichloroacetate, in terms of their inhibition or accentuating effects, or activating effects, I guess would be a better word. But the real thing to keep in mind is that a buildup of acetyl-CoA and NADH increase PDK, which further shuts down pyruvate dehydrogenase, while acetyl-CoA buildup and NADH all in and of themselves also shut down pyruvate dehydrogenase. So you have multiple mechanisms here shutting down pyruvate dehydrogenase activity with a buildup of NADH and acetyl-CoA. And this is going to be really important when we go through the graphs, and I'll explain all of that. The last thing I just wanted to read here is inhibition of glycolysis. So pyruvate dehydrogenase is the enzyme that links glycolysis to the Krebs cycle. So that's the main block, that linkage enzyme. But the glycolysis, as I discussed before, also gets inhibited, but the inhibition is smaller compared to the inhibition at pyruvate dehydrogenase. So fatty acid oxidation doesn't really shut down glycolysis as much as it shuts down glucose oxidation or through the Krebs cycle. And so what they're just saying is there's about a 20 to 30% decreased uptake for glucose due to an increase in fatty acid oxidation. So this would be at hexokinase. And then there's a 40 to 60% decrease flux through phosphofructose kinase 1. So that's the flux, I think it's of glucose 6-phosphate through this enzyme to create, I forget the other product that it creates in the glycolytic pathway, but it basically will block this enzyme or decrease the flux of the product of the substrate through the enzyme by 40 to 60% and decrease that glucose uptake specifically by 20 to 30%. And again, this is compared with almost complete inhibition of pyruvate dehydrogenase. And then it says, as initially proposed, phosphofructose kinase 1 inhibition results from the accumulation of citrate, a well-known allosteric inhibitor of phosphofructose kinase 1 and phosphofructose kinase 2, as later demonstrated in liver and heart. So an allosteric modulator, I know it sounds like a fancy word, but essentially it's when something binds onto an enzyme, or perhaps, it binds onto an enzyme and basically impairs that enzyme's ability to undergo its reaction. So the way I guess you could think of it is like if the enzyme is this like crystalline structure or like a series of magnets, like a ball of magnets with different interactions amongst each other, and that's giving it its shape, kind of like if you had like those magnetized marbles and you throw them on the floor, they'll all interact to create this shape. If I put a super strong magnet on one side, it changes the shape of those magnets. And now if there was a little pocket in those magnets that was supposed to bind the glucose 6-phosphate, the glucose 6-phosphate doesn't fit so well anymore because the citrate, in this case, that bound onto the phosphofructose kinase changed that pocket shape. And now the molecule, the glucose 6-phosphate essentially doesn't fit as well, so the reaction doesn't occur as nicely or as strongly or as good as we want it to. So that's essentially what an allosteric modulator is. It's just something that binds to an enzyme and impairs its function. I know it has a fancy name, but that's basically what goes on. So now, basically, we've gone through, this is the meat of the article. I'm not going to go too much more because it gets into like PPARs and genetic control of these different things. I really just wanted to talk, and that's separate from the Randle cycle. I wanted to just talk about the Randle cycle, but I want to describe the process for you guys here. So what I was describing earlier is, I don't have glycolysis pictured, but with glycolysis, you have one molecule of glucose creates two molecules of pyruvate. And then you have the enzyme. I don't know if you guys can see it here. This is, let me see. So there's pyruvate dehydrogenase. So you can see that there. The pyruvate dehydrogenase is the enzyme that takes the pyruvate from glycolysis and turns it into acetyl-CoA. And by turning it into acetyl-CoA, it gets, eventually there's another reaction with citrate synthase where citrate is created from oxaloacetate and then the acetyl-CoA. So this is a cycle that continues to spin, and then feeding glucose in the cycle that in the form of 2-pyruvate allows the cycle to occur. Fatty acids can also enter into the Krebs cycle through acetyl-CoA. I'm pretty sure it's acetyl-CoA, yeah. So then, and that allows the energy production to occur. Now, what you'll see here, and now the one thing that's not pictured is the FAD and FADH2, but it's similar to, if you guys see here, there's NAD plus and NADH. So what you're seeing when you see these little arrows coming off is NAD gets fed into this reaction of isocitrate dehydrogenase. So you get isocitrate plus NAD plus moves through the enzyme isocitrate dehydrogenase and forms NADH and an H plus ion, otherwise known as a proton. So these enzymes are producing the NADH. There's also around here, around the succinate dehydrogenase, the production of FADH2. I'm pretty sure it happens around succinate dehydrogenase. So succinate plus its FAD moves through succinic dehydrogenase or succinate dehydrogenase and then produces the FADH2 amongst other components. I don't memorize them off the top of my head, but essentially this is really important to understand because when you have carbohydrates, you get a higher production or a higher ratio production of NADH to FADH2. So the carbohydrates produce more NADH in ratio to FADH2 than fats do. Fats produce, comparatively to carbohydrate, more FADH in ratio to NADH. And this will become a really important process as we discuss later. When I go through as you can see behind here, there's the little the electron transport chain. But essentially, those electron carriers are very important. So in the process of beta oxidation, where you're basically harvesting the fatty acids and the carbons off the fatty acids and their bonds to create these electron carriers in the form of NADH and FADH2, you get, comparatively to carbs, more of that FADH2 than NADH. And then with carbohydrates, you have glycolysis, and then they also move through the Krebs cycle. You'll get, comparatively to fats, more NADH than FADH2. I'm not going to go through exactly why that happens now. Perhaps I'll make a video on showing the actual breakdown and the count and the ratios of FADH2 and NADH2 between beta oxidation and glucose oxidation. But just right now, all you need to know is that the ratio is higher with fatty acids of FADH2 to NADH compared to carbohydrate. And that becomes extremely important when we go to the electron transport chain, which we're going to go to in a second. Now, the only other thing I want to share with you here, just to keep in mind, is that in the Krebs cycle, if you notice, in order for this reaction to occur through isocitrate dehydrogenase or pyruvate dehydrogenase or alpha-ketoglutarate dehydrogenase or malate dehydrogenase, you need NAD. You need NAD. So you need, for example, if you want to create alpha-ketoglutarate, you need deisocitrate and NAD for this reaction to occur. If you don't have NAD, this reaction cannot occur because you don't have the precursors for it to occur. Same thing with pyruvate dehydrogenase, specifically pyruvate dehydrogenase, and we'll get here in a sec. So just keep that in mind. You need NAD. You also need FAD. It's just not pictured here. So now let's go over to the electron transport chain. Here we have the electron transport chain. You can see, let me just move it up a little bit. Here you have the lipid bilayer. So this is the phosphate head with the fatty acid tails. Then you have the intermembrane proteins. These are the complexes: complex 1, complex 2, complex 3, complex 4. They're, I know it's very creative names for them. Other thing I want to point out here is you have coenzyme Q, and this is extremely important. We're going to talk about it in a second. So essentially what happens at the electron transport chain, these carriers that we produce in the Krebs cycle, NADH and FADH2, and the reason I call them carriers again is because they're holding electrons. So NAD plus can accept 2 electrons, then it becomes NAD without the plus because the electron's negative, and the negative electron cancels the positive charge of the NAD, and then it gains another electron, which makes it NAD minus, which allows it to hold a hydrogen ion or a proton, because the proton is positively charged. So now you have NADH, which has a neutral charge because the electrons and the protons are canceling each other out. And the same thing happens with FADH2. So you have FAD, it gains 2 electrons, and then it can hold 2 protons. So that's why you have FADH2. Now the electrons from NADH and FADH2 move to their respective complexes here. So NADH drops 2 electrons off at complex 1, and FADH2 drops 2 electrons off at complex 2. Now, in order for these electrons move to different enzymes, and as they move to these different enzymes, they pump these H plus ions across the membrane. So the enzymes use the energy from the electrons, kind of like, little pumps, and they pump these H plus ions that you can see right here into the inner membrane space of the mitochondria. And what happens is there's less H plus ions here, and there's more H plus ions over here, kind of like a battery. Now, essentially what you see, there's another enzyme that's not listed here. It's called ATPase, and you can think of it like a hydroelectric dam, like a water wheel. And as these H plus ions come through and they move down through the enzyme, you can literally say this isn't ATPase, but say this was ATPase. They just move down through ATPase here, and as they turn that little water wheel, you get ATP is created. I guess it's the most simple way to think of it. Now, essentially what you're seeing is that the electrons are powering these complexes, but they're moving from complex to complex. So the electrons at complex 1 go to coenzyme Q, which carries them to complex 3. The electrons at complex 2 go to coenzyme Q, which carries them to complex 3. Those electrons can move through cytochrome C and then to complex 4. Now, you have there's a nuance here, there's a caveat, and some of you may have caught it. Complex 1 and complex 2 both share coenzyme Q. This is a big deal because if you have a lot of FADH2 here, complex 2 starts to hog coenzyme Q. So the electrons are going to complex 2 and then coenzyme Q, and complex 2 and then coenzyme Q. So what winds up happening is NADH can drop its electrons off at complex 1, but there's nowhere else for them to go. So complex 1 starts to get jammed up. And what happens from the complex 1? You start to see a large production of reactive oxygen species from these electrons with nowhere to go, nothing to do. The other thing that happens is when you get jammed up here, NADH cannot unload the electrons to complex 1, and so you start getting a backlog of NADH. So if you have too much FADH2 in ratio to NADH, which is what happens, again, with fatty acid oxidation, with beta oxidation, you get more FADH2 in ratio to NADH comparatively to carbohydrate. What winds up happening with that change in ratio, it leads to excess FADH2 at complex 2, which basically hogs coenzyme Q, creates this backlog and issue at complex 1, where you build up NADH and you produce ROS. Now, the ROS can be a problem because the ROS can damage this membrane, can damage the proteins, can damage the mitochondrial DNA, etc. So what happens? Now, it depends on how much ROS is generated, but an uncoupling protein can come to the membrane, and the theoretical basis essentially will basically dissipate the H plus ions in the form of heat. So another protein gets put to the membrane. It's called an uncoupling protein. The reason it's called uncoupling is because it uncouples the electron transport chain from the Krebs cycle in the sense of producing energy in the form of ATP. What winds up happening is it just increases heat production. So the uncoupling protein comes to the membrane here, basically eliminates this H plus ion gradient, and basically helps to minimize the ROS production. Now, what winds up happening is you don't have ATP generation because you're minimizing the gradient that generates ATP, and you have this buildup of NADH and then the ROS, which also, again, the ROS starts to get dissipated to a large extent through the uncoupling protein. The uncoupling proteins are there to minimize ROS and also to generate heat. So in situations of stress or excess ROS generation, uncoupling proteins are literally there to dissipate the ROS to protect the mitochondria in certain situations. Now, so again, we have this buildup. Now what we're left with is decreased ATP because the uncoupling protein is there because of the ROS. Maybe we have mitochondrial damage if our mitochondria membrane is full of polyunsaturated fats, and we have this excess of NADH. So what happens when we have this excess of NADH? So we're going to come back over here to the Krebs cycle. Now we have the Krebs cycle is producing this NADH for us, right? But what does it need to produce the NADH? It needs NAD+. So we're basically building up this NADH, and we don't have enough NAD+ to run these reactions. So what happens? The system gets backlogged here, right? So you're going to get a buildup of these different metabolites because there's not enough NAD+ here to run isocitrate dehydrogenase or pyruvate dehydrogenase. So acetyl-CoA gets backed up here. So you get this excess acetyl-CoA to CoA. You get a buildup of citrate all the way back. And then you also get a buildup of NADH with nowhere to go. So what happens? The buildup of NADH, the buildup of acetyl-CoA shuts down pyruvate dehydrogenase, and then the buildup of citrate goes and shuts down the enzymes in glycolysis, the enzyme in glycolysis phosphofructose kinase 1, and then also that buildup at phosphofructose kinase 1. That buildup of its substrate, which is glucose 6-phosphate, then can shut down hexokinase, which can impair the movement of glucose into the cell because you have this buildup of the glucose 6-phosphate and nowhere for this to go. Or you have somewhere to go because there's the phosphofructose kinase 1 is still working to some extent, but its function has been strongly decreased. So you basically get a buildup there, but you have this huge inhibition of pyruvate dehydrogenase and this buildup of acetyl-CoA. So what are you left with essentially? You can get a buildup of pyruvate. So now you can't oxidize carbohydrate effectively through the Krebs cycle. You have a decrease of ATP generation at the cell to some extent. If there's a large amount of ROS and uncoupling proteins had to be increased, you have an increase in ROS, and then you have impaired insulin sensitivity because you have that whole backlog through. This is the Randle effect. This is essentially the Randle effect. This is what happens when you have an excess of fatty acid oxidation, and a large part of it stems around that ratio again of FADH2 to NADH. That with fatty acid oxidation, there's a comparatively higher ratio of FADH2 to NADH than there is in carbohydrate oxidation, and it causes this whole backlog. And basically, you get stuck oxidizing fatty acids because pyruvate dehydrogenase gets shut down. And then essentially, if you do have a buildup of pyruvate, then that buildup of pyruvate could possibly go to lactic acid fermentation and produce a smaller amount of ATP from carbohydrate than you would if you had fatty acid oxidation. Obviously, glycolytic flux gets decreased as well. So this is the Randle cycle. This is the fatty acid oxidation, inhibition of carbohydrate oxidation. These are the mechanisms here. It's what happens at the electron transport chain and what's going on at the Krebs cycle. And again, it stems to that FADH2 and NADH ratio. It's extremely important. Now, last thing I want to talk about here is what does this actually look like in research studies. So what they say is in mouse models, the genetic suppression of pyruvate dehydrogenase in cardiac and skeletal muscles results in dramatic effects in male pups, which die on weaning, but not in female offspring. These pyruvate dehydrogenase deficient female mice can survive if fed a high-fat diet, but they then go on to develop muscle hypertrophy and heart dysfunction. In contrast, upregulation of pyruvate dehydrogenase kinase by genetic manipulation or in response to high-fat diet, starvation, or insulin deficiency keeps glucose oxidation at a low level, whereas fatty acid oxidation is increased. So in both these states, if there's they have mice which have had their genes adjusted, or they've played around with their genes so that the pyruvate dehydrogenase in their muscles basically has a significantly decreased function. The male mice will die, but the female mice, essentially, they can only eat a high-fat diet, and they'll be able to live. But over time, they develop muscle hypertrophy and heart dysfunction. So this muscle hypertrophy, this isn't a good thing. This is actually a response to poor energy production at the muscle cells. They try to increase the muscular fibers, or perhaps there's a buildup of metabolites within the muscle cells, which could form the hypertrophy. And you also get heart dysfunction. And I think this occurs because, as I was talking about, when you have an excess of fatty acid oxidation without the glucose oxidation, what winds up happening is you can get at a certain threshold a large amount of ROS. This large amount of ROS can lead to upregulation of the uncoupling proteins and basically a decrease in the production of ATP. So you have an energetic dysfunction, and then a large amount of ROS can also damage the cells and damage the mitochondria. So over time, the high-fat diet allows them to survive, but it doesn't allow them to thrive because they're still stuck with an energetic deficiency and or damage to the mitochondria from an increase in ROS because they can't effectively oxidize enough carbohydrate. Again, there's thresholds here, so it's not just if you're oxidizing fat at all, it's bad, because the heart does oxidize fat to a large extent, but it's ratios. It's how much fat is the heart oxidizing versus carbohydrate, etc. So there's some nuance to the picture, but essentially inhibiting carbohydrate oxidation leads to severe issues. The other thing they talk about is basically inhibiting upregulating pyruvate dehydrogenase kinase. Now again, you can do it through genetic manipulation, or you can just create a high-fat diet, undergo starvation, or have insulin deficiency, which could be like a state like diabetes. This could be intermittent fasting. This could be keto or carnivore. And essentially, now there's to some extent, if you're doing carnivore and it's very high protein, this may be altered a bit because of an excess of gluconeogenesis. So even if you're high fat, you may still have some carbohydrate, but there's more problems with this, and it's tangential. But essentially, this will help keep glucose oxidation at a low level, and then fatty acid oxidation decrease. Now, they go on to say that this situation mimics a state of metabolic inflexibility, essentially a failure to adapt metabolism to the fasted to fed state transition, which is a feature of insulin resistance. Again, this situation mimics a state of metabolic inflexibility. Essentially, let me just turn the light on. Essentially, when you have an excess of fatty acid oxidation due to an upregulation, or if you have an excess of fatty acid oxidation, and which can cause an upregulation of pyruvate dehydrogenase kinase and impair glucose oxidation, this could be caused by high-fat diet, starvation, or insulin deficiency. You enter a state of metabolic inflexibility where you're forced to oxidize fatty acids. So you're literally metabolically inflexible. This flies in the face of some of the comments I've seen by low-carb supporters or low-carb gurus who say that oxidizing fatty acids and minimizing carbohydrate oxidation leads to a state of metabolic flexibility. This is just basically it's wrong. It's basically incorrect, because the excess fatty acid oxidation through the mechanisms that I just went through, and also, again, I'm not even saying this, what the researchers are saying here is it creates a state to some extent of insulin resistance and an inability to oxidize carbohydrate effectively, which is the metabolic inflexibility. So again, shutting down pyruvate dehydrogenase either genetically or upregulating pyruvate dehydrogenase genetically, or by increasing high fat feeding, inducing starvation, perhaps fasting, and then also insulin deficiency, creates a state of metabolic inflexibility and insulin resistance. And in at an extreme level with blockage of pyruvate dehydrogenase, you basically get death in the offspring of the animals that were genetically modified to have that deficiency, or like muscular failure to a large extent with hypertrophy and dysfunction over the long term with the high fat feeding. So again, this is a feature. This is what happens on extreme levels in these situations with shut down of pyruvate dehydrogenase. Now, say if we upregulate pyruvate dehydrogenase, what they say here is conversely, the blood glucose level in starved pyruvate dehydrogenase kinase 4 deficient mice is lower than in wild types, probably because the active pyruvate dehydrogenase diverts pyruvate, a gluconeogenic substrate, to acetyl-CoA. These observations underlie the crucial role of pyruvate dehydrogenase in glucose and lipid homeostasis. So essentially, what they're discussing here is that if you downregulate pyruvate dehydrogenase kinase, which is, again, that's the enzyme that kind of blocks pyruvate dehydrogenase. So if you're downregulating this enzyme, you upregulate pyruvate dehydrogenase. And in doing so, it directs the products from glycolysis, which is pyruvate, to becoming acetyl-CoA and entering the Krebs cycle. And that basically, number one, that essentially would increase insulin sensitivity to some extent. So you would have more glucose being able to flux through the Krebs cycle, but it also can minimize the gluconeogenic substrate, which is pyruvate, which can basically be turned into glucose and put out into the bloodstream by the liver. It basically moves it into being oxidized through the Krebs cycle. And again, these examples here, they say these observations underlie the crucial role of pyruvate dehydrogenase in glucose and lipid homeostasis. So these are just going to show the mechanisms that I just described, what actually happens in real physiology if you alter this enzyme's function in mice or rats. What you wind up seeing is if you decrease pyruvate dehydrogenase heavily, you're forced into fat oxidation. And if you upregulate pyruvate dehydrogenase heavily, you're forced into glucose oxidation. So pyruvate dehydrogenase is that switch, and it's also the spot where the Randle cycle is through various mechanisms affecting its switch. So if you have an excess of fatty acid oxidation, pyruvate dehydrogenase is getting shut down, and then carbohydrate oxidation and uptake by the cells gets shut down, kind of through a backlog effect to the cells. So that's essentially it for this paper. That was what I wanted to cover for the Randle cycle. Hopefully this was helpful for you guys. If I would maybe watch the bioenergetic helpline number 8 and then come to watch this video so it can give you a background, or maybe watch this video before you watch bioenergetic helpline number 8 so you can understand the arguments and what we're discussing with Bart K, about Bart K's video. Again, it's just about concepts, nothing about him personally, just literally about the Randle cycle. So yeah, if you guys like this type of content, if you like these videos, go ahead, give me a thumbs up, give me a like, etc., whatever works for you. And if you have any feedback on the videos, please let me know, because obviously I'm kind of just getting into the producing the YouTube videos myself, so I'm learning as I go. All right, guys, hope you enjoy the content. Stay tuned for some more. Next videos, I think I'll go over the Bohr and Haldane cycles.